I'm Dr. Sabrina Siegel with the Neuroscience Education Institute, and twice a month I sit down with a renowned mental health care expert to discuss breakthroughs and best practices for treating patients with mental illness. In this episode of the NEI podcast, we are interviewing Dr. Donna Wilcock, a Sweeney Nelms professor in Alzheimer's disease research and the associate director of the Sanders Brown Center on Aging. Welcome, Dr. Wilcock.
Hi, really nice to be with you today.
Thank you. Dr. Wilcock, could you share with us the relationship between vascular cognitive impairment and dementia?
Yes, so this is an area of intense research right now because we're really realizing that the vascular component of the brain, the cerebrovascular system, is extremely sensitive to damage, and that any damage to that cerebrovasculature can impact the way the brain functions. And so when we talk about dementia, we're really talking about a clinical syndrome. Alzheimer's disease, we know, is the leading cause of dementia. But actually, a lot of Alzheimer's patients have some cerebrovascular dysfunction and disease also associated with those Alzheimer changes. And so when we talk about vascular cognitive impairment, we're talking about a cognitive impairment that is due to some kind of damage to the vasculature, whether that is a small stroke, a large stroke, a small hemorrhagic event. It could even be blood-brain barrier leakage in certain areas. Hypertension can damage the very small vessels in the brain. And all of that can lead to a cognitive impairment that can contribute either on its own or in association with other neurodegenerative processes like Alzheimer's. It can lead to the culmination of a clinical dementia syndrome. Depending on which paper you read, anywhere from about 60 to 80% of Alzheimer's cases will have some vascular component to their disease as well. And so with that high prevalence has come a lot of interest in trying to study the vascular disease in the brain, because a lot of our clinical trials, all of our clinical trials really to this point in Alzheimer's disease have failed. And so a lot of us are going back and questioning why those trials have failed when we've really been very successful at treating our mouse models of Alzheimer's. And one of the big differences is that our mouse models have a very pure disease. And our patients that we're rolling these treatments out to don't have just plaques and tangles of Alzheimer's in their brain. They have a lot of other processes going on at the same time. And the most common one of those other processes does seem to be this vascular disease of some type.
You sort of touched on this already, but how prevalent is vascular impairment in patients with Alzheimer's disease?
Yeah, so a lot of the estimates depend on what you are defining as vascular impairment. And so if you're just considering overt cerebrovascular pathology like a stroke or like multiple microinfarcts, let's say, the prevalence of that is probably in the area of around 20% of Alzheimer's cases. If you broaden that out to say on a microscopic level, which ones of those have some vascular change or disturbance, whether that is microinfarcts, microhemorrhages, blood-brain barrier leakage, white matter dysfunction. Once you start incorporating all of those as being your vascular impairment, you end up with an estimate closer to that 60 to 80% number.
Wow. What are some of the molecular mechanisms of vascular impairment that your research focuses on?
So my lab has been focused for some years now on studying the inflammatory component of vascular disease. So when you have any kind of oxidative stress damage to the vessels, or when you have some blood-brain barrier leakage, that will activate the microglia that are sitting close to the vessel in what we call the neurovascular unit. And so the neurovascular unit is the blood vessel and its surrounding cells that help that vessel function. So astrocytes have contact with blood vessels. Neurons do also have some input onto those vessels. You have the smooth muscle cells, you have pericytes, and you can have microglia around the vessels. So any kind of damage, acute damage to the vessel will activate the microglia. What we found is that activation of microglia initiates a process that leads to more blood-brain barrier breakdown, because you have proteinases now that degrade the tight junctions that form the barrier. And also, we've now seen that this inflammation can lead to the degradation of the astrocytic end feet around the vessel. And so now you actually have what appears to be a retraction of astrocytic end feet from around the vessel. And so when that happens, you lose some of your capacity to regulate blood flow and also regulate the water. The osmotic homeostasis gets dysregulated. And then you seem to get into a vicious cycle then of you get surrounding tissue damage, and the neurons are not maintaining their resting membrane potentials. And so it's kind of a downward spiral from there. So inflammation has been really a key part of our research in the lab for a long time. More recently, we're seeing some changes in proteins responsible for angiogenesis, and angiogenesis being the growth of new blood vessels.
Mm-hmm.
We're just starting out really looking at this, and it's based on some data we've got from our patient samples. But in the retina, there is a process called pathologic angiogenesis, and it occurs in cases of macular degeneration in the retina, where you get a lot of new vessels being formed, but they're very immature and they're leaky. And so we actually think that you may be getting some of that pathologic angiogenesis occurring in the brain. And it's the body's normal response to a hypoxic event. So if you have some kind of small infarct, or you have some damage to the local capillaries, stopping them from being perfused, that you have a local hypoxia that kind of signals that, hey, we need to get more vasculature going in here because we're not getting enough energy, enough nutrients. And so this angiogenic process starts, but it's not tightly regulated enough. And so you end up with these immature, non-perfusing, potentially vessels that probably lead to more and more damage, probably through inflammation at the end of the day. So a lot of it always keeps coming back to inflammation.
Right. And that was actually going to lead me to my next question, which is, what is the role of inflammation in vascular impairment?
Yeah, so it's interesting. Really, the answer here is we don't know.
Mm-hmm.
And that's why we need to get a lot more research going on this. What is very interesting is that a lot of the inflammatory immune phenomena that has been studied in stroke seems to actually have the opposite effect when you have a chronic cerebrovascular dysfunction. Things that have been shown in stroke to be reparative actually seem to be detrimental in our models of chronic cerebrovascular disease. So there's definitely a difference between an acute response and a chronic response. And probably in the acute setting, these are beneficial responses. But in the chronic setting, those beneficial responses become detrimental and actually cause more tissue damage. So there's been a lot of instances, not just for us, but for other labs that are exploring some of these pathways, where there's an assumption that you're going to see the same things that you see in stroke, and that targeting the same pathways as stroke will be good. But actually, when we take it back to the bench, we don't see that at all. We're seeing, very opposite outcomes. So, the short answer is we don't know what the role of inflammation is. But the longer answer is that we really need to not make assumptions based on what we've seen with acute cerebrovascular disease. And actually, we have to study these pathways in the chronic condition.
What is the influence of cerebrovascular disease on the progression of Alzheimer's disease? What do we know about that at this point?
Yeah, so that is interesting. And some of the best work has come out of Julie Schneider's group at Rush University, who have very elegantly shown in their clinical cohorts that the time to dementia is shortened when you have cerebrovascular pathology with Alzheimer's pathology. So really, for a given amount of Alzheimer's change, so let's say you have plaques and tangles of a certain level, and if all you have is those plaques and tangles, you don't have a dementia. But if you have that same amount of plaques and tangles and you also have cerebrovascular change on top of that, you will most likely present with dementia. But what they've shown is that once you have the symptoms, the rate of decline is very similar. Whether you just have the plaques and tangles of Alzheimer's or whether you have that plus vascular, if you track the rate of decline in an individual, it seems to be quite comparable. It's just that your threshold for dementia is decreased with the vascular pathology.
What are some of the biomarkers that you're studying that might be indicators of cognitive impairment due to cerebrovascular pathologies and Alzheimer's disease?
So that's really interesting. We are a part of a consortium that has been funded by the NINDS and the NIA to not only identify new biomarkers for vascular cognitive impairment, but to also validate those biomarkers among the consortium. And so that's really important because there's a lot of reports out there about one particular biomarker or another particular biomarker. It's usually out of one group and it doesn't necessarily get replicated. And so the goal of this consortium is to identify potential biomarkers, but then among the 7 sites that are part of this consortium, for the most part, it's all 7 sites, are actually validating those biomarkers within their own cohorts to show that they have value. I think that's really important. The biomarkers, and the consortium is called MARC VCID, so MARK VCID. And the biomarkers range from, there's several that are MRI based, and one is looking at, for example, cerebrovascular reactivity. So what that means is measuring blood flow in the brain, but then in the MRI using arterial spin label, but then actually challenging the patient with CO2. So you hook them up to a mask and you increase the CO2 levels and you actually cycle that CO2. With that cycling of the CO2, you should see in the brain an increase in blood flow and a decrease in blood flow in response. And the time it takes for the brain to increase that blood flow in response to the CO2, that's actually called cerebrovascular reactivity. How quick that vasculature is to react to this hypercapnic challenge. That's one biomarker because that reactivity appears to go down with age and with cerebrovascular disease. Another biomarker that we're studying is an angiogenic protein called placental growth factor, PLGF. And this is one that raises some eyebrows with that name. Lots of people, kind of look at you like you've got two heads when you talk about placental growth factor. But it's one of these unfortunate instances in biology where it's a protein that was named for where it was discovered and not necessarily what it does. And so it is definitely involved in the placenta during pregnancy, and it's been studied in preeclampsia, but it is more globally expressed. And we have seen the levels of this angiogenic protein go up both in the spinal fluid and in the plasma in our patients that have vascular pathology as evidenced on MRI. So that is a biomarker that we are currently cross-validating among the sites of Monk VCID.
Wow.
To see whether that will be a biomarker. And, we've got kind of fingers crossed on that one. But I think that also points to potentially therapeutic targets.
Yeah, which that actually leads me to my next question. But that's all so exciting, and thank you so much for sharing that with us. With that being said, what therapeutic approach does your research focus on?
So it really focuses on the two pathways that we have talked about, that being inflammation. And so we are exploring targeting inflammation with vascular models, but the neuroinflammation landscape has become so much more complicated in the last few years than it used to be. Any type of targeting of inflammation these days is not just trying to globally knock down inflammation, say with an NSAID, because that seems to inhibit the beneficial aspects of inflammation as well as the detrimental aspects. What we're trying to do is target specific immune pathways that we know have been associated with vascular damage. So one of the pathways that we're looking at is TNF alpha. That's a cytokine that's a pro-inflammatory cytokine. There's different therapeutic approaches used for rheumatoid arthritis, for example, that already target TNF alpha. So that's one thing that we're starting to look at in our models. The other pathway that we're very interested in is because of the biomarker data that we have on the placental growth factor, is trying to study PLGF as a potential therapeutic target. In the mouse models, we can get a knockout mouse for PLGF, which is what we're in the process of trying to get now. And if we can get that PLGF knockout mouse, we can do our model in the mouse and we can see if they're protected from damage. If they are, then that really points us to this being a modifiable biomarker, and that perhaps neutralizing antibodies could be one approach to lowering PLGF. Or maybe if we can find out why PLGF is being increased, we can try and target upstream of PLGF and target whatever is driving it. So that's a little bit more in the early stages than the inflammation is, but I do think it could be a real potential approach.
Wow, that all sounds so exciting. Thank you so much for sharing all of this great information with us, and thank you for being on the show.
Thank you for having me. Thanks so much.
This podcast was brought to you by NEI Learn. Access all of NEI's CME activities from your mobile device. NEI Learn is available for download on iTunes or Android.